Good morning, friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic fashion. I hope this will be very convenient for the continuous study. Thank you very much, my dear friends. We have been started with a new series of leukemia. In that series, in the last class, we have discussed about some of the basics of leukocytosis, and one of the important differential diagnoses for leukemia, that is leucomoid reaction. When you are looking into the CBC, leucomoid reaction, how it happens, and these details we have seen. And lastly, we have also covered some of the types of proliferation which takes place in the bone marrow lymphoid cells. So two types of leukocyte proliferation will take place. One is called as leukocytic proliferation and another one is neoplastic proliferation. When I say leukocytic proliferation, that is taking place under normal limits. It follows all the normal rules and it is well controlled by the body. That division or proliferation of the leukocyte which is taking place inside the bone marrow lymphoid series, so that is under well control of genetic engineering, what do you say? Okay, it's a genetic control, genetically mediated, and it is well controlled cell division of the leukocyte. It follows the rules. So when you talk about the cell division, whatever may be, it may be leukocyte or anything, normal body cells, skeletal cells, there is a rule. Okay, so telophase, anaphase, prophase, the rules are there. According to that rules, there will be division of the chromosomes and all, and that follows the rules. That should be the pattern in normal. Then proliferation to differentiation. Normally when proliferation takes place, it starts from the proliferation and then differentiation. Differentiation means as the proliferation takes place, the quality of the cells will go on increasing. Okay, so it follows the rules. Okay, for example in RBC, so we need to undergo differentiation. What happens is initially there will be nucleus, then the size will be very large, there will be nucleus, then later onwards the size become smaller and smaller, nucleus disappears, hemoglobin comes. That is a normal rule, okay, that has to be followed, and this is called as differentiation. Same thing also takes place in the leukocytes. So a normal leukocyte proliferation, it follows this rule. Then it starts from the immaturity to the maturity. Initially the cells were blastocytic cells, precursor cells are that means initially they are immature cells and later they get into the mature form. They release and produce only when required. So their release or their proliferation is stimulus mediated, in the sense whenever there is a need and whenever there is a call from the immune cells, these leukocytes will go into start proliferation, and then they release into the blood. So this type of normal leukocytic proliferation takes place in normal whenever a person encounter with any type of infections, etc., as per the call of the immune system. But whereas when we talk about neoplastic proliferation, this is different from the leukocytic proliferation. What is the difference? Neoplastic proliferation is not controlled. Controlled in the sense cell division has got its own time, and as I told, it's gene mediated process. There should be release of some stimulation, some of the genes will produce some of the ribosomes, that ribosome will produce the related proteins. Like that, things are totally under control. But when I talk about neoplastic, there is no rules followed. Okay, cell is dividing by its own without any stimulus, without any control, without any command. Okay, so the diffuse way of distribution, and the distribution is also very diffuse. And important is proliferation takes place, and that is proliferation is not leads to differentiation. Rather, proliferation will go to lead to de-differentiation. What I mean to say is, the next series of the cells which is formed from the parent cell, okay, so qualitatively it is worse than the parent cell. Okay, so therefore there is no differentiation, there is de-differentiation. So that particular cell is totally waste cell because it is not able to perform any expected functions, because it does not contain such cell organelles inside it, because it is undergoing dedifferentiation. Suppose in 4 mitochondria should be there in a cell, but no. When the division takes place, there is only 2 mitochondria, endoplasmic reticulum is absent, like that. Previously, the contents which are present in the cell are not present in the daughter cells. Okay, so that means it's because it is uncontrolled, uncommanded, and unexpected divisions. Okay, therefore proliferation will not lead to differentiation, rather proliferation leads to dedifferentiation. So this leads to immaturity of the cells. As the proliferation takes place, the cells become more and more immature and useless. There is no maturation also. So no stimulus for the production, that genetic breakdown will be there. So that means stimulus is not there and things are not taking place according to the genetic signaling. Okay. So this is the difference between the neoplastic proliferation and the leukocytic proliferation. Now, leukocytosis, neoplastic proliferation, we will look into the details because that will be our topic. Now, this is another type of proliferation which takes place in the bone marrow house stem cell, as I explained, because all this neoplastic proliferation of the leukocyte that will take place in the bone marrow stem cells. In this case, the normal pattern of the leukocytosis production is not found. Normal pattern, what is that? We have already seen. So that is not found. Cause may be different. So why this neoplastic proliferation takes place? Why normal leukocytic proliferation will not take place? Why it is shifted to the neoplastic proliferation? Causes are many. According to some, the genetic cause is very important, and according to some, there may be some acquired causes, there may be some drugs, there may be some trauma, etc. Infections mediated. Many things are there which disturbs this normal pattern of leukocytosis and shift it into neoplastic proliferation. Its proliferation will not take place as per the demand, as I explained earlier. It is not taking place as per the demand, rather it is totally uncontrolled. Okay, uncontrolled proliferation should take place according to the need. No, without any need also, your stem cells are go on dividing without following any commands. Okay. Now, the leukocytes are released from the stem cells and then they spread in a diffuse manner and spread throughout the bone marrow. So what happens? Initially the leukocytes are released and that is abnormally formed leukocytes are released, and these cells are diffusely spread in the total bone marrow. Then these cells are not following the rules of differentiation. As I told earlier, it is not following the rules of differentiation, rather it also follows the dedifferentiation. Okay. Now they remain big and not undergo the metamorphic changes. Now as far as when I say neutrophils, initially neutrophils contains many lobes. That many lobes has to be limited to 7 or 9 lobes at the end as they go on maturing. But when they mature also, if the number of lobes are same or increasing rather, then it is not called as differentiation, it is called as dedifferentiation. Okay, so they remain bigger, their size should come to, for example, RBC previously it was very big, but at the end it should reach the 7 micron. Now this size has to go on reducing. If it is not happening, then it is not called as differentiation, it is called as dedifferentiation. So same type of things also takes place in the leukocyte. Okay, so they remain bigger and not undergo the metamorphic changes. And these cells are now very too sticky in nature. Okay, whenever they go and settle, they'll stick there. So they are also not freely flowing or flexible. They are very sticky in nature. And some of the cells which are diffusely spread on the bone marrow, sometimes they leak out from the bone marrow and entering into the circulation. Some of the cells. And those cells which come into the circulation, now they reach the other lymphoid tissues in the body through the blood. It may reach the lymph tissues, it may reach thymus or some other spleen, etc. Usually lymph glands, so they will take it. Now these leaked cells from the bone marrow, when they come to the circulation, they enter into the different parts of the body. Usually they are taken by the lymph nodes and now they get settled in the limbs. Okay, lymph nodes or lymphoid tissues. They get accumulated there. So initially there is only 1 or 2 cells. They come and get in a particular lymph node. There they now start proliferation. Abnormal proliferation now started in the lymph nodes. And as a result they form the node-like structure. And this is called as lymphomas. Now this is the basic difference between leukemias and lymphomas. Leukemias are limited to the bone marrow. Lymph nodes So lymphomas are nothing but the same leukemic cells which are leaked out from the bone marrow and get settled in the lymph nodes. So the diffuse proliferation when occur inside the bone marrow house, it is known as leukemia. Okay, so usually it is spreading, diffusely spreading and sticky in nature, therefore it is called as milky wave pattern. And the same when it occurs in the other extended areas of the body like lymph gland, then it is called as lymphomas. So lymphomas just like butter from the milk, how you can collect the butter. So butter will be sticky compared to the milk, and there is a node-like structure. So these cells are very sticky and differentiated and will not do any normal function. Totally they are abnormal cells doing an abnormal function, and they are dangerous to the body. If they are remaining dormant, then also no problem, but they are acting in a negative way and more immature. So immature. So now these particular de-differentiated cells which are abnormally dividing, so they are not performing any normal body functions. Okay, now when it is not performing any normal function, we cannot call this a cell. Okay, rather it is now called as blast. These abnormal cells which are de-differentiated, which are formed without any reason, which are totally immature, which are de-differentiated, dividing without any control, not following the body command, they are not called as cell, rather they are called as blast. Now this is how the blast forms inside the bone marrow. If undergo proper differentiation at the end, it is called as cyte or cell. Now it is not cell or cyte, it is called as blast. So now it is called as not leukocyte, rather it should be called as a leucoblast. Okay, so the differentiation now it begins with the blast. So when such phenomena occurs in the lymphoid series, suppose it is called as lymphoblast, and when it occurs in the myeloid series, it is called as myeloblast. We know that myeloid series produce neutrophils, basophils, etc. So when it happens in the myeloid series, it is called as myeloblast. When it happens in the lymphoid series, it is called as lymphoblast. Its production is totally uncontrolled, so it goes on producing the blast without any stimulation, which is immature, large, and with a less quality, sticky cells, both inside the bone marrow and outside the bone marrow in the form of lymphoma. So then it is called as leukemia. Okay, very simple, I repeated many times. So clinically what we find is abnormal increase in the leukocyte. When you are doing the CBC, clinically what you find is abnormal increase in the leukocyte, which should be differentiated from the normal leukocytes and leukocyte reaction. I will order for a CBC. CBC shows increased WBC cells, increased leukocyte. Increased leukocytes, now whether it is a normal leukocyte or abnormal leukocyte, this will not be expressed in the CBC report. CBC report says that leukocytosis, that may be increased. Suppose the child is having some infection and all, normal cell pattern is taking place, normal leukocytosis is taking place. Yes, there may be increase up to 20,000, 25,000 acute infections. Okay. Even in case of leukemia where the leukoblasts are abnormally proliferating, that also shows leukocytosis in CBC. So when we have got leukocytosis, we should differentiate it from one, normal leukocytosis, and second, I already told in the first class, that is leukocyte reaction. Now there will be abnormal LAP, this leukocyte alkaline phosphatase level will be seen. This is, in the last class also I told how to differentiate. Okay, suppose if it is leukemia, towards leukemia, then LAP is increased, leukocyte alkaline phosphatase is also increased, and on the peripheral blood picture we find lots of immature cells of lymphoid series with the blast cells. As the day passes, this abnormal series of leukocytes overcome the normal series of the leukocyte production. Now, previously these abnormal leukoblasts are very few in number, but as the day passes, they take over the normal series of leukocyte. Okay, in the beginning only few are there, later they continue because they are abnormally dividing, abnormally increasing. Okay, so now they are increasing, and one day comes where they overcome the normal series production of leukocyte, and bone marrow is fully 90% of the bone marrow is totally with the abnormal blast cells. So when the blast cell production is more than 20%, okay, if the blast cell production is more than 20% of the total bone marrow stem cells, then it is termed as leukemia. Okay, so if in the lymphoid tissue it leads to lymphoblastic leukemia, and if it is in the myelin tissue it is called as myeloblastic leukemia. And this is the precursors lymphoid in the thymus. First lymphoblasts are produced. That lymphoblasts, they undergo B cell, T cell and natural killer cells. Okay, and there will be a stage of rapid proliferation of the lymphoblast, the stage of switching off of the proliferation and switch off of the differentiation. So like this, these are the normal things. Now we come to the another part. What do you mean by acute and chronic leukemias? Now when there is proliferation, as we know in normalcy it follows the rules, while leukemia it will not follow any rules and it is totally out of control, everybody's control, it is out of everybody's control. So this arrest in normalcy may be due to various causes as explained. This arrest may occur in various levels. It may in the very beginning or it may be at the end. I will explain this. More the level of the arrest from the normal proliferation, less the chance of severity as the cells become more matured. Okay. Chronic lymphocytic leukemia, as rapidity of the production of the blast cell is less, whereas in acute leukemias the rapidity of production of the blast cell will be more. Early the arrest of the normal proliferation, more will be the rapidity of the production, very less will be the maturity, and the chances of severity is more. So then it is called as acute lymphoblast leukemia. Now I will explain you. When the precursor cells are there, when precursor cells are there, they are undergoing rapidly dividing. Okay, so they are when rapidly dividing, the proliferation is taking place. In the blast cells, what happens? So during this rapidly proliferating stage, the blast cells are rapidly dividing. Okay, now if this rapidly dividing phase of proliferation, if its arrest takes place earlier, then the possibility of getting chronic leukemias is more. But if the rapidity is rapidly progressing, that is rapid proliferation is rapidly progressing, then there will be more and more production of the leukocytes and chance of producing acute leukemia is more. So this acute leukemias and chronic leukemias, this actually basically depends upon at what point this arrest takes place. If the arrest takes place in the earlier part itself, then less blasts are formed and more normal cells will be there, and the possibility is leukemias get converted into chronic type. But all of a sudden all the blasts are proliferating at once and arrest is not taking place. Okay, more and more immature cells are formed, then chance of getting the acute leukemias is more. It's just like I give an example of brainwash in the later stage. That means a child, for some reason he has been brainwashed in the early childhood itself, then he will be more and more dangerous. Suppose your brainwash has been taken place after 50 years. That means when you are already mature. Okay, you are already mature. Now you are able to differentiate which is good and which is bad. In that type, somebody come and do your brainwash, you will not be so easily get into their trap. Okay. Similarly here also, that means if the arrest takes place in the early part itself, so you will have more mature cells. Okay, that mature cells will not lead to such a abnormal proliferation. But If the arrest takes place in the later parts, that means in the first part of chronic proliferation, the arrest takes place in the later parts, that means already your brain has been brainwashed, you have been brainwashed, then its control is very difficult. So similar is the case. This is how the acute and as well as chronic leukemias begins. Now in the next class onwards, we shall discuss in detail about leukemias. Thank you very much.